Our next speaker is Murti Salapaka from the Electrical and Computer Engineering Department, and I'll ask Vivan Sharma to prepare for the next talk.
Oh, thank you for inviting me here. So I'm going to motivate this by a disease which is essentially Duchenne muscular dystrophy, which affects 1 in 5,000 males. And there's no cure for this particular disease. It's a wasting disease which it affects boys, and usually they have a lifespan of 20 years with degrading health. And the main mechanism because of which this disease affects these individuals is because of this molecular protein called dystrophin. Um, and this dystrophin molecule is essentially provides support to this the sarcolemma, the cell structure. When you are moving your muscle, it provides this cushioning effect so that the cell structure integrity is maintained. And kids who don't have this dystrophin molecule suffer from this disease because they don't have dystrophin itself. So my colleagues in the College of Biological Sciences, Jim Arvasti, he was one of the first to have purified this protein molecule that I've shown in the bottom here with domain structures here that you see here for various spectrum type repeats. And clearly, this is a mechanical protein in the sense that it provides mechanical stability to the entire cell structure. But nobody had studied this from a mechanical point of view, and so we studied it. My expertise towards this. So these proteins are, typically the forces are in the piconewton range, so very small forces. And what you see here is a representative force profile. And we would like is to design this domains, what sequence of domains will lead to a required force profile, for example, for dystrophin, and then we could design other proteins that are easily integrated into a human cell so that it can have a force profile which looks like to that of dystrophin itself. So this is a bigger project, but I just wanted to bring out that the importance of machine learning. And what we do is we typically take a protein molecule here, and what you're seeing is the protein molecule is stuck to a probe here, other end to a substrate, and we basically pull this protein molecule. And as the protein molecule domains unravel, it pops. Each of these domains unravels, it pops and provides a force profile. And what you're seeing is this protein molecule tension increasing, going down when one of the domains unravels, and you get a particular force profile of this particular protein molecule. And that's the mechanism by which it provides this shock absorption of a muscle movement inside a cell. It's very difficult to do these experiments, and because you have to isolate the protein and do all these experiments one by one. And also much more confusing is this cantilever can actually attach to multiple proteins, not just one protein. And so you have to decipher between, is the data coming from a single protein? Is it coming from multiple proteins? How many proteins? And then go on and doing the inferences on the molecule, on the data itself. And so what we have done is, I'm not going to go into the methods of how we do machine learning on this, but we have automated this process. Another step is actually looking at different domains, and these are heterogeneous molecules, so they have different domains, and automatic classification of the domains in the data itself. And there also, again, machine learning becomes quite useful. So stop here and take questions offline, yeah.